Okay, so we're going to go ahead and complete these population genetics problems. Again, you should have tried these problems before now, and you should really just use this video as a check of your work. Make sure you got everything right before you submit it for your grade, but it'll also help you in studying for the exam. Okay, so let's get started. So question number 1, we'll read it and we'll work through it. So a liver disease is caused by a recessive allele. Okay, so you have a deleterious recessive condition causing liver disease. And again, that's caused by a recessive allele. 1 in 100, so 1, oh, that's not gonna work, here we go. Get my pen. 1 in 100, no, it's still not gonna work, I gotta use my finger. 1 in 100 people have the condition, okay? What are the P and Q values of this population, and what is the percentage of carriers? So let's start off first by trying to figure out P and Q values. So there's 2 questions here. So 1 in 100, what does that equal? Okay, that's going to equal our Q squared. So remember our 2 formulas. P plus Q equals 1, P squared plus 2PQ plus Q squared equals 1. Remember what each of those represents. So P is the frequency of the dominant allele, Q is the frequency of the recessive allele, P squared is the frequency of homozygous dominant individuals, 2PQ is the frequency of heterozygous individuals, and Q squared is the frequency of homozygous recessive. And so here we have the numbers that we can use to figure out the frequency of homozygous recessive individuals. So Q squared is equal to 1 divided by 100. Okay, so then if you do the math, 1 divided by 100 is 0.01. Okay, so that's Q squared. That's the frequency of the homozygous recessive condition. In this case, it's the frequency of liver disease in a population. I want P and Q. So I can figure out Q by taking the square root of both sides. So the square root of Q squared is Q, and the square root of 0.01 is 0.1. So I have Q. Okay, now to find P, I use my formula. P plus Q equals 1. So 1 minus Q will equal P. So 1 minus 0.1 gives me P. That's my dog, sorry, which is 0.9. So there, I've got my P and Q values for this population. So there's the answer to the first question. The second question says, what is the percentage of carriers in the population? Okay, so as soon as you hear carriers, you should think 2PQ. So carriers are those heterozygotes. They don't have liver disease. However, they carry that recessive deleterious allele that codes for liver disease. So they are carriers of that nasty allele. Okay, so now you just plug in your P and Q. So this would be 2 times 0.1 times 0.9. And hopefully I did my math right. That should equal 0.18. So that's the frequency of carriers, but it's asking me for a percentage. So I want to make sure I convert that. So multiply it by 100, and I get 18%. Okay? So that's the answer to that first question. Okay. Moving on to the second question. Okay, so this says the frequency of alleles in a gene pool is 0.1 for capital letter A, that's the dominant allele, and the frequency of the recessive allele is 0.9. So we have P and Q. So P, sorry. Okay. Why is that not letting me do that? Hold on a second here. Sorry. Okay, so P equals 0 and Q equals 0.9. Okay, so then it says what is the percentage in the population of heterozygote individuals? It's worded terribly. I should say what is the That's fine. What is the percentage in the population of heterozygous individuals? Sorry about that. So heterozygotes are 2pq, which is going to equal 2 times 0.1 times 0.9. Okay, so 2 times 0.1 times 0.9. Oh, same. It's going to be 0.18. Again, they're asking for a percentage, so we're going to make that 18%. Okay, so there's the first question. The next part of the question, remember when you're doing these genetics problems, make sure that you box in your answer so that I don't have to search for it. Okay, I can just kind of check it. The next part of the question is, what is the percentage in the population that is homozygous recessive? Okay, so same P and Q values. Get my eraser here. Okay, so let's erase this. Hopefully. Keep our P and Q values there because we need those. Okay, and we're looking for again, the percentage of homozygous recessive. So homozygous recessive, hopefully you know, is Q squared. Okay, and so that's going to be 0.9 squared. So 0.9 squared is 0.81. And again, that's the frequency of homozygous recessive individuals, but we want a percentage, so you got to multiply that by 100, and that's going to give you 81%. Okay, so there's the answer to that second part of that question. Okay, so moving on to the third question. Okay, allele B for white wool is dominant over allele small b for black wool. In a sample of 900 sheep, 891 are white, 9 are black. Estimate the allelic frequencies in this sample. Okay, so you can figure out the frequency of the homozygous recessive individuals. Okay, let's start there. So there's 900 sheep, 9 of them are black. Okay, so the frequency of the homozygous recessive individuals, or Q squared, which is where we like to start these kinds of problems, is equal to 9. Where did I get that? 9 black sheep. They've got to be homozygous recessive. If they're black, they must be little b, little b. And there's 900 total sheep. So 9 out of 900 are homozygous recessive. Get my calculator out here. So 9 divided by 900 equals 0.01. Okay, that's Q squared. Well, I want Q. So I'm going to take the square root of Q squared as well as the square root of 0.01. So that's going to be Q equals, I'm going to type it, 0.1. Okay? And if Q equals 0.1, then what does P equal? 1 minus 0.1, which is 0.9. So there's my P and Q values. Those are the allelic frequencies in this sample. Okay, that one was pretty easy. Let's go on to number 4. So number 4 says, in a population that is in Hardy-Weinberg equilibrium, so they're in stasis, it's not evolving, this population, the frequency of the recessive homozygote or homozygous genotype of a certain trait is 0.09. Okay, so that's going to be Q squared equals 0.09. They want to know the percentage of individuals homozygous dominant for the allele. So I've got to get P squared. Okay, so how am I going to get that? I need to get P first. Okay, so I'm going to take the square root of Q as well as the square root of 0.09. So Q equals, let's see, 0.09 square root is 0.09. 0.3. That means that P must be 0.7, right? Because P plus Q has to equal 1. And to get the percentage here, I've got to get P squared, which is going to equal 0.7 squared. So let's do that. 0.7 squared equals 0.49. So P squared equals 0.49. Well, they want the percentage, so we're going to have to multiply 49 by 100, and that's going to be 49%. Okay? And that's it for that one. So that was number 4. Moving on to number 5. Okay, so number 5 reads, in a population that is in Hardy-Weinberg equilibrium, 36% of the individuals are recessive homozygotes for a certain trait. Okay, so right there, 36% is equal to 0.36. And that is the frequency of the recessive genotype. So that's Q squared. Okay? So now the question is, for the same trait, what is the percentage in this population of homozygous dominant individuals? So we know that 36% are homozygous recessive. How about homozygous dominant? So I need to find P, because I got to get to P squared. So I'm going to take the square root of both sides of this. So that's going to be Q is going to equal 0.6. The square root of 30.36 is 0.6. And so if Q equals 0.6, then P equals 1 minus 0.6, which is 0.4. Okay, so there's the frequency of my alleles. Now I'm just going to do P squared to get the frequency of my homozygous dominant individuals. So that's going to be 0.4 squared, which equals 0.16, right? Yep. So 0.16, but they asked for a percentage, so I got to multiply that by 100, which gives me 16%. So that's the percentage. This is the frequency of homozygous dominant individuals. The last part of this question, it wants to know the frequency of the heterozygous individuals. So, can I move myself? Yeah. So heterozygotes, I got to do 2, where was I? Here, okay. 2PQ. So that's going to be 2 times 0.6 times 0.4 equals 2 times 0.6 times 0.4 equals 0.48. And of course we want percentage, not frequency. So that's going to be 48%. And we should be able to add up those 3 percentages, hopefully, to get 100. So 48 plus 36 plus 16. Yeah, my calculator's playing tricks on me. So yeah, 48 plus 16 plus 36 equals 100. So our math adds up there. Okay, so that was question number 5. So let me move myself back to the question. Okay, so question 6 says allele T, capital T, codes for the ability to taste a particular chemical, and that's dominant over allele little t, which codes for the inability to taste. Okay. At Cornell University, out of 400 students surveyed, 64 were found to be non-tasters. Okay. What is the percentage of heterozygote students? Okay, so you can look right here. Those non-tasters, those are homozygous recessive, right? Little t, little t. And so we got 64 out of 400, so that's going to be our Q squared. our frequency of homozygous recessive individuals. So that's going to be 64 divided by 400. Okay, we'll do that math real quick. Maybe, yeah. Okay, that equals 0.16. So Q squared equals 0.16. So how do I get Q? I take the square root of both sides. And so the square root of 0.16 is 0.4, right? Yeah, 0.4. So I got Q. How do I get P? 1 minus Q. So P is going to equal 0.6. Okay, so now I have my P and Q. I can go on to figure out my frequency of heterozygotes. So you know the formula for heterozygotes, right? It's 2PQ. So 2 times 0.4 times 0.6. 2 times 0.4 times 0.6. And this is not on purpose. This is the same numbers as last time. Didn't do that on purpose. So it's going to be 0.4%. So we want to make sure that we convert that into 48%. Well, there's my answer. So that's question number 6. So, question 7 here. Oh, okay. In humans, Rh positive individuals have the Rh antigen on their red blood cells, while Rh negative individuals do not. Assume that the Rh positive phenotype is produced by the dominant allele. Okay, so dominant allele gives you Rh positive, and the negative phenotype is produced by its recessive allele. Okay, so recessive allele, negative. In a population that is in Hardy-Weinberg equilibrium, so not this particular gene is not under any selective pressure. If 84% of the individuals, okay, so 80, keep doing that. Get my pen back. Okay, so 84% or 0.84 are Rh positive. Okay, so that is the dominant phenotype, okay, which is going to be made up by homozygous dominant and heterozygotes. So 84% are homozygous dominant and heterozygous, together. So that's, how are we going to do that? We want to know what the frequencies of the dominant and recessive allele are, so P and Q. We can't use 84% directly because that takes into account the frequencies of both of those genotypes, so that's a problem. We could, however, figure out from that the frequency of the homozygous recessive phenotype. So how do we do that? We just do 100 minus 84. So 16% of the population, or a frequency of 0.16, are going to be homozygous recessive. So 0.16 equals Q squared. Okay, same number again. So again, we take the square root of both sides, and so Q equals 0.4, and P will equal 0.6. Okay, know what you're looking for. So that was question number 7. Okay, question number 8 is a little bit long. And so what I did here is I've got the page of questions, and then I've got, I think, a blank page for us. Yeah. Okay, let me just make sure I have my pen. Okay. So the question reads, 2 pirates and 3 Polynesian beauties settle on an uninhabited tropical island. Ooh, we have the founder effect. All 5 settlers had brown eyes. Okay. But only one man carried the recessive allele for blue eyes. Okay, so we got 5 people, right? And we're going to use big B and little b. So big B is going to be the allele coding for brown eyes, and little b will be the allele coding for blue eyes. And so we've got 5 settlers that all of them have brown eyes, but one man carries. So he's going to be the only heterozygote. So here we go. We got big B, big B. There's 1. big B, big B. There's 2, big B, big B. There's 3, big B, big B. There's 4, and our 5th settler is going to have this genotype. Okay, so those are our 5 people, our 5 colonizing people. So the first question says, part A, it says, what are the P and Q values for this pioneering group? We can go right off of alleles here, right? So we've got 2, 4, 6, 8. We've got 10 total alleles. Only one of them is recessive. So this is kind of easy. We could do, I'm going to write right here, Q equals 1 divided by 10. Okay, and P equals 9 divided by 10. Okay, so Q is going to equal 0.1, and P is going to equal 0.9. Okay. So, out of room here. So, equals 0.1, and P equals 0.9. So there's our first answer. So for A, it says, what are the P and Q values for this pioneering group? Our value for P, which is the frequency of the dominant allele, is 0.9. And the frequency of Q is 0.1. Or we could say, out of those alleles, 90% of them are dominant and 10% of them are recessive. Okay, either way. So let's go on to B. Now for B, I'm going to need the next slide. So it says, the population expands to 1,500 individuals, and it's in Hardy-Weinberg equilibrium. So what that means is that the P and Q values haven't changed. So P is still 0.9, and Q is still 0.1. And it asks how many blue-eyed individuals would there be in this expanded population. Okay, let's go to the next slide. So remember the frequency, I think I wrote it over here, of Q is 0.1. So Q equals 0.1. And we want to know how many blue-eyed individuals. So we need to know the frequency of the homozygous recessive genotype. So that's Q squared. Q squared. Okay, and so to get Q squared, what's our Q 0.1? Squared is going to be 0.01. Okay, so that's the frequency of the homozygous recessive genotype. Now, there are how many individuals? 1,500. We want to know how many out of that 1,500 are homozygous recessive. So that's going to be 1,500 times 0.01. And that's going to give us 15. So that is the answer to 4B. There are 15 individuals that have blue eyes. Okay? So that's that part. Let me erase that. Okay, so let's back up one slide and let's read C. So we got our P and Q values in A. In part B, we figured out that there was 15 blue-eyed individuals. And so in part C, it says how many brown-eyed individuals. Okay, so brown-eyed individuals are going to be P squared plus 2PQ. We got to figure out the frequency and then the number of individuals of both of those genotypes. So the homozygous dominant as well as the heterozygotes. Okay, so first off, P squared equals 0.9, I believe, squared. Okay. So 0.9 squared equals 0.81. 81. And we got to multiply that by 1,500 individuals. That's going to give us 1,215. So that's how many homozygous dominant. And then 2PQ, we're going to do 2 times 0.9 times 0.1. And that's going to give us 0.18 times that 1,500 equals 270. So we need to add these two together to figure out the number of brown-eyed individuals. And that's going to be 1,485. And we can check our work if we add the 15 blue-eyed individuals. Yeah, we get 1,500. Now, you could have gotten that in a much easier way, right? You could have taken your total population of 1,500 and just subtracted how many blue-eyed individuals there were, which was 15, and you would have gotten 1,485 brown-eyed individuals. But I wanted to show you how to get it using p squared and 2pq. Make sure you know how to do that. Okay, so we answered question A, B, and C. So here's the tricky part. In part D, back up a slide, it says if all of the blue-eyed individuals, so 15 people, died at the same time, what would be the new P and Q values of the following generation? Okay, so now this violates Hardy-Weinberg equilibrium. Okay, we're going to get allele frequency changes because we just wiped out all of the homozygous recessive individuals, and so those frequencies are going to change. Okay, however, the number, not the frequency, but the number of P alleles is not, because we didn't wipe out any brown-eyed individuals. Okay, so keep that in the back of your mind while we work through this. Okay. So let's erase everything on here. Okay, so first off, let's think about that original population. There were 1,500 individual people in that population. So how many alleles? Well, all of those people are diploid, having 2 alleles for every gene. And so if we do the math there, that's 3,000. So in that original population, there were 3,000 alleles. I'm not spelling too good here. Okay, so 3,000 total alleles, okay, for this particular gene, eye color. Okay, and so in that original population, let's figure out how many of those 3,000 alleles were brown alleles. So we're going to do 3,000 times the original frequency of P in that population, which we know was 0.9 in the original population. And so if I do that math, that's 2,700. So that's the number of brown alleles. And remember, we only are losing, in part D, we're only losing blue-eyed people. So we didn't lose any brown alleles, only blue alleles. So the number of brown alleles that we had in the original population is still going to be the same. The frequency isn't, because we now have 30 less alleles. Okay? So let's figure out how many alleles now there are in the remaining population. So we could either do 3,000 minus 30, or you could use your answer from part C, which was here we go, 1,485. So we said that 1,485 people remained after we lost the 15. We got to multiply that by 2 to get the number of alleles. And that's going to be 2,970. Okay, which is the same thing as 3,000 minus 30. So either way, you could have done it. So here's the total number of alleles left after we lose all those blue-eyed people. Here's the number of brown alleles that are present. So now we want to know the frequency of the brown alleles. So all we do is we take the number of brown alleles, 2,700, and we divide that by the total number of alleles, 2,970. And that's going to give us the frequency. So if you do that math, that's going to give us our P value, which is going to be about 0.91. It's like 0.90999. So 0.91 is close enough. So that's going to be our new P value. So it's a slight shift. We only lost 15 people, so it's going to be slight. So how do we get Q? We do 1 minus 0.91, which is 0.01. So those are our new P and Q values. So yeah, there's a shift. Okay? All right, so that's that one. Okay, so question number 9 is another long one. So again, I've got a nice blank page following. Okay, so it says 4% of a population suffers from a serious debilitating disease. The condition is caused by a recessive allele. Okay, so there we go. We start off with Q squared. So Q squared equals 4% or 0.04. Okay, so let's do question part A right on here. So Q squared equals 4%, which you know is equal to 0.04. How do we find Q? We just take the square root of both sides. And so Q is equal to 0.2. And if Q equals 0.2, then P has to equal 0.8, right? How do I know that? 1 minus 0.2 is 0.8. So that first part was nice and easy. Okay? Gets a little trickier. Make sure I have my calculator on. Okay. Next question. It says how many carriers? Okay, as soon as you see carriers, what are you thinking? You're thinking 2PQ, right? Our carriers are our heterozygotes. So how many carriers are in a population of 2,500? So first I have to find the frequency of carriers, and then I can find the number. So let's go to the next slide. So the frequency of carriers. So I got 2PQ, which equals 2 times 0.2 times 0.8. And so that should equal 0.32. So the frequency of carriers is 0.32. The percentage of carriers is 32%. But I want to know the number, right? Because it doesn't ask you for a percentage. It says how many carriers. So I have to have the number of carriers. So how do I do that? I take the total population, which is 2,500 individuals, and I multiply that by the frequency of carriers, which is 0.32. And hopefully you got 800. So out of my population of 2,500 people, 800 of them are heterozygotes or carriers. They carry this recessive, deleterious, nasty allele that causes this serious, debilitating disease. Okay, let's go back. So that was the answer for 9B. So 9C, this takes us back to our genetics from chapter 12. It says if 2 carriers had children, what are the probabilities of their having offspring with this serious condition? Okay, so don't overthink this. It just says 2 carriers had children. So I'm going to use the letter A to represent this allele. So I have 2 carriers, so 2 heterozygotes. That's all it is, right? Just do a Punnett square. So let's do it on the next page here. So let me erase this. Okay. So big A, little a times big A, little a. Punnett square. So I got a big A allele and a little a allele. Pardon that noise. That's my husband making me a wonderful dinner. Here's my Punnett square. Just 4 boxes. So in my first box here, I'm going to put big A, little a, big A, little a, and little a, little a. Okay, so what was the question? It said, if 2 carriers had children, what are the probabilities of their having offspring with this serious condition? So 1/4 or 25% of their offspring will be born with this serious, debilitating condition. Okay? All right. Last part of that question, it says, after repeated generations of selection, would you expect the value of Q or the frequency of Q to drop to 0? Will it be wiped out? Why or why not? And this is kind of neat. In lab, you're going to do an exercise just like this. You're going to take a population of rabbits, and the rabbits are going to either have red or white fur. And white is dominant to red. And for generations, there was no advantage of having either red or white fur. Rabbits of either coat color survived at an equal rate. Okay, so remember again, white is dominant to red. And then all of a sudden, well, first of all, these rabbits are going to be living in the Arctic tundra, so on a nice white background of snow and ice. And then one day, a new invasive species of hawk is introduced, and it's a visual predator. And so it's going to attack and eat any rabbits that it can. And so those red rabbits, obviously on that white background, are going to be pretty easy prey, where the white rabbits will be camouflaged. So they're going to have an adaptive evolutionary advantage. And so we're going to see allele frequencies change over, I think it's 6 generations you're going to take it out to. And the question is will we lose the red allele, which is the Q allele, the recessive allele? So it's the same idea here. And it's kind of neat. You're going to see that because recessive alleles, even if they're debilitating, because they're hidden in heterozygotes or carriers, they're usually not wiped out from a population. Okay, and as you do that lab, and I really want you to do the lab, don't just make up numbers, you'll see that you're going to take it out to 6 generations. You'll see that those alleles are not wiped out, and you can actually make a pretty good prediction of whether or not the allele can be potentially wiped out ever. Okay, and usually we'll find that the answer is no. Because it's hidden in the heterozygote, the allele will not be lost. Okay? Okay, so last question here. Okay, so we've got a population of humans, and we've got tall and short. Okay, so being tall being tall is dominant to being short. So the tall allele, I can't get my pointer on. Okay, there we go. The tall allele is dominant to the short allele. Okay, so big T equals tall and little t equals short. So we've got a group of individuals, and this is going to be another example of that founder effect. They're going to leave their population, and they're going to go to a deserted island, so a new area, and they're going to colonize that new area. So who's going? We've got 2 short women. Okay, so remember, tall is dominant to short, so those short women are going to have the genotype little t, so there's one of them. Here's the second. And then we've got a short man, and they're going to be with 3 heterozygous tall men. Okay, so there's all the people. So there's 1, 2, 3, 4, there's 6 of them, or 12 total alleles. Okay, so let's find the P and Q values first. So let's do P. Okay, so P is going to equal the number of tall alleles, which is 3, divided by the total number of alleles, which is 12. And then we can do the same thing for Q. Q is going to equal 9 over 12. And so the frequency of P is 0.75. So if we do that math, it's 0.75. And the frequency of Q is going to be 0.25. Okay, so there's the answer to the first question. So let me get rid of a little bit of this ink so that we can see. I should have just hit erase all. Grab my pen back. Okay, so P again is going to equal 0.75 and Q equals 0.25. Okay, good. So there's A. So B says, after several years of inhabiting the island, the population grows to 2,500 individuals. If Hardy-Weinberg conditions are in effect, so that means P and Q values are the same, how many homozygous tall, heterozygous tall, and homozygous short people would you find in the population? Okay, so we got 20, so let's do our P and Q up here. So P equals 0.75, and Q equals 0.25, and our population is 2,500 people. And we want to know first how many homozygous tall people there are. Okay, so that's going to be P squared, which equals 0.75 squared. So 0.75 squared equals about 0.5625 times the total number of people, 2,500. Okay, so remember, so this number right here is the frequency of the homozygous dominant genotype. And we're going to multiply that by the total population to figure out how many people are homozygous dominant, tall. And that's going to be 1,406.25, which you can't have a quarter of a person, but we'll keep it like that. Okay, so that's how many homozygous tall people there are. That's part of that question. Then it asks you to find, I think, the heterozygotes next. Okay, so the heterozygotes, wait, I did that in the opposite. I switched my P and Qs. That was the number, did I switch them? Yeah, I switched them around. Sorry, hold on, let's fix this. So P should be less, should be 0.25, and Q should be 0.75. Just trying to see if you're paying attention. So if we come up here, again, P equals 0.25, and Q equals 0.75. Okay, so now P squared, let's start this one over, equals 0.25 squared, which equals 0.0625. Now you multiply that by 2,500. That makes more sense. And that's going to give us about 156.25 people. Okay? So that's going to be my number of homozygous dominant individuals. Okay. So now let's do the next thing on there. So the next question says, look, heterozygous. So that's going to be 2PQ, which is equal to 2 times 0.25 times 0.75. Okay, and that should equal 0.375. Again, times that entire population, which is 2,500. And that's going to give us about 937.5 people. So that's the number of carriers, or the number of heterozygotes, in this population of 2,500 people. So there's just one more to get. And that's going to be my number of homozygous recessive. So that's going to be Q squared, which is going to equal 0.75 squared, which is equal to 0.5625. Multiply that by 2,500 people, and that is going to give you 1,406 people. Well, 406.25 people. Okay, so we just figured out the number of homozygous dominant, the number of heterozygotes, and the number of homozygous recessive individuals. So now there's just one more part left to this question. It says, and this is just like the last one, or the two ago. And there are questions like this from lecture, and there will be some questions this week when we do this lab in the lab. Okay, so it says, a decree is handed down, so a rule is handed down from the governing body, which rules that all short people, okay, so that's the last one that we just did, so that was 1,406.25 people. They gotta go. They have to leave the island because it's getting too crowded, so they're just gonna nix them. What are the new P and Q values of the remaining population? Okay, remember, here's your hint. The number, not the frequency, but the number of P alleles in the original population and the new population is the same. We didn't lose any tall alleles. Okay? So, let's erase this. This is tricky. This is the third one that we've done like this. There's one in lecture. Okay, and you're going to see one in lab too. So you want to make sure you know how to do this. And who knows, maybe on a quiz or a test. Okay, so we're going to start off. Where am I? Let's start off with the original population. Okay, so in the original population, we had 2,500 people. And people are diploid, so we're going to multiply that by 2 to get the total number of alleles, 5,000, in the original population. Okay, now the question is, how many of those alleles were big T, dominant alleles? Okay, so we're going to take our original P, which was 0.25, and we're going to multiply that by those 5,000 alleles. And if I did my math right, that should be 1,250 alleles. So that's how many P, or I should say big T alleles there were in that original population. And that's how many big T alleles there still are in this population because we didn't lose any tall individuals. We only lost short. So we're going to use that number in just a second. Now the question now is, how many alleles are left in the population? So we got rid of all the short people, and from question part B of this question, we knew that that was 1,406 people. Hold on a second. Let me get my eraser. So we started off with 2,500 people. We lost 1,406.25 people. So how many people remain in the population? So if you just do that subtraction, it's going to be 1,093.75 people. Great. How many alleles is that? So we got to multiply that by 2. So 1093.75 times 2. This is going to give us the number of alleles. The number of alleles left in the population. So just do that math. That's 2,187.5. Okay. So that's how many alleles are left. This is how many alleles are dominant alleles. So if we want to know the new frequency of P, we take the number of dominant alleles, 1,250, and we divide that by the total number of alleles left in the population. And that should equal about 0.57. So that's your new p-value. To get Q, you just do 1 minus 0.57, so that's going to be 0.43. It's tricky. Okay? So again, the more you practice, the better you'll get at this. This is the third, I think, question of this type that we had. There's one question in lab that's kind of like this, but it's actually the reverse. I think we wipe out all of the dominant phenotype individuals, which is interesting. So what would happen? So let's do the reverse of this. So if in C, if I instead said to you, there's a rule that wipes out all tall people, what are the new P and Q values for the remaining population? You would say to me, well, if you wipe out all of the tall people, that's all the P squares and all the 2PQ, so all you're left with is homozygous recessive short people. And so that's really easy. P becomes 0, there's no T alleles, and the frequency of Q, the little t alleles, becomes 1. So that would be really easy, so be careful. There might be a question worded like that in lab. Okay, so again, hopefully you are getting the hang of doing these types of problems. You'll have more practice in lab. There'll be a couple on the quiz, a couple on the exam. Probably nothing this difficult. But anyways, you should be able to figure these out. So hopefully that helped. So again, make sure that after you've checked your work, that you go ahead and you upload your completed, double-checked assignment right to Blackboard so that I can give you credit for that.